Today we'll discuss another important technique that we use regularly in molecular genetics labs and molecular biology labs called gel electrophoresis. Some of you may have heard of this technique before. Well, no, some of you may have even used it before, but we'll go through some of the theoretical framework for it, and we'll introduce it today because we are going to refer back to it quite a bit through the end of the semester. So we left it at conversation about how PCR can have several uses. In fact, when I asked what we use PCR for, I heard some of the ideas that are listed on the screen now. But we also emphasize that PCR as a molecular biology technique, all it is a protocol or a procedure by which we can get many copies of a specific DNA sequence, right? And of course, having many copies of a specific DNA sequence can be useful in a number of ways, and that's what we'll do today. Go through some of these in complete lack of order. We'll start with the third one. So one of the most common uses for PCR is to find out if a given DNA sequence is present in a sample or not. And my question to you is, how can we use PCR to determine that if a sequence of interest is present in a sample? What would you do?
If it doesn't, it wasn't?
That's right. Yeah, very simple, very straightforward. If we're interested in finding out if a given DNA sequence of interest is present in a sample or not, we subject that sample to a PCR reaction with primers that would recognize that sequence of interest. And if we get many copies of it, it was. If we don't, it wasn't, right? Now, that's very easily said. The problem we run into is that PCR will make millions or billions or trillions of copies of your small DNA fragment of interest, but it will also make it against a backdrop of a lot more DNA that was present in that original biological sample, because biological species have genomes that far exceed in size the typical size of your PCR amplicon, right? So what happens is, yes, in your tube before the PCR you had a lot of DNA. Yes, after the PCR you have a lot more DNA. But if you were to use some sort of technique that shows presence of DNA in an attempt to try and find out if your PCR reaction worked or not, there is a good chance that the amount of DNA that you see in both before and after the PCR may be comparable, simply because you're not able to detect the many millions of copies of your very small fragment against a backdrop of a few copies of gigantic genomes. That always presented a problem, and part of the solution actually, in a way, preceded the use of PCR, but the solution came to mind really quickly for people who work with DNA in labs at the time that PCR was invented. And that is, what we know about these PCR fragments is that they should be of a specific size. And now we go to what you were suggesting before. If we could take a look at whether or not we have many copies of DNA pieces that are of that specific size that we anticipate, then we can more confidently determine if the PCR worked or not. So if we look at, the question that we ask is there DNA in my sample after the PCR? We'll always see DNA. Now, if we ask, are there many copies of a fragment that is of this particular size that I know I should get if the PCR worked, then the answer to that question is much more meaningful. So how do we determine what may be the size of different fragments of DNA present in a sample? And that's where gel electrophoresis comes in. So for those of you who never heard of the technique, I'm assuming that some of you never heard of this before, so I'll walk you through the whole thing slowly. Gel electrophoresis is a technique that is based, as the name indicates, on using a very special kind of gel. When it comes to nucleic acids, DNA and RNA, the gels that we normally use are made of a compound called agarose, and it's not much different from Jell-O, the Jell-O that we eat, right? So, and it is, these gels, as the one shown here in the picture, are made the same way that you would make Jell-O to any shape of a container that you wish, right? So you have a powder, the agarose in powder form, you mix it with water, you heat it up to really high temperatures, that agarose will melt, then you let it cool down. When that agarose cools down, it will form molecular bonds between different agarose molecules, such that from a macroscopic point of view, it jellifies, so it forms that gel to the shape that you used in this case. We call this a gel bed, and it's just a plastic space that will contain the gel in a rectangular form. And on a microscopic view, what we would see is that all those agarose molecules created these intramolecular links between them, giving rise to pores that are of different size depending on how much agarose you had in your gel. So the higher the concentration of agarose, the smaller the pores are going to be. And these pores end up forming a 3D mesh, or a 3D sieve, if you will, that will work as a filter for molecules, in this case, different DNA fragments, and it is pretty intuitive. The smaller the DNA fragment, the easier it will be for it to go through these pores. The larger the DNA fragment, the more slowly it will go through them. It will advance through them because it can squeeze. Imagine that even long DNA fragments are ultimately like a string, so you can always thread a string through a small pore, but it will take a little longer. Right? So hold that thought for a second because it'll come handy later. After we've solidified our gel, we would place it in a tank that is filled with what we call an electrophoresis buffer. It's just a aqueous solution with some electrolytes. And when we solidify the gel, we use these special combs such that the agarose solidifies around the teeth of these combs. And when we take the combs out, those teeth leave these indentations in them that we call wells, the wells in the gel. And we will use those wells, those pockets, to place our DNA samples. In this image, you can see some of these wells are occupied by some blue liquid that indicates the presence of a DNA sample in them. Right? So not all wells have been loaded here. A few of them have. All right, so without knowing much more, you can probably appreciate that DNA will be in these wells on one end of this gel. And I told you that this gel is like a 3D mesh that offers some resistance to different molecules of DNA of different size. So smaller DNA molecules will have lesser trouble going through those pores. Larger DNA molecules will have a little bit of a harder time. Now, how can we force DNA to move through these pores? The use of electricity. And that's, if you will, the second part of the name of this technique. Electrophoresis means pulling with electricity, right? So what we do is we make use of the negative nature of DNA as a molecule. And what we do is we subject these tanks that are filled up with liquid to an electrical current, such that on one end of that tank, there's going to be the negative pole, and on the other end, there will be the positive. I didn't choose the location of these two randomly. Notice that DNA, being negative, is on the same side as the negative, because what we'll want is that DNA to move or migrate towards the positive. That's where DNA will want to go when we turn on this electrical current. So DNA will move through the gel to the positive, and that's how we accomplish size separation, because we're subjecting all of these DNA molecules to a pull with an electrical current towards the positive end of the tank, but the smaller molecules will have an easier time going through those pores in the gel than the larger molecules. Pretty intuitive, right? Any questions so far? No? Cool. So, what we do is we let the samples run through the gel. There are ways we have to make sure that they don't run out of that gel, that they don't come out on the other side of the gel. So we try and keep the samples within that slab of the agarose gel. And after we've run those DNA molecules some distance, we can reveal their presence using special chemical dyes that will bind to DNA and have the unique characteristic of fluorescing under either ultraviolet light or some type of special light, typically UV light, right? So the way this works is the samples run through the gel as far as they could, given their size, and then we subject these gels to a staining with a molecule that will bind to DNA, and wherever they bound DNA, and when we hit them with UV light, they will emit some fluorescence light that we can see with special cameras or sometimes even by with a naked eye. So this is the typical cartoon showing a gel in it's coming up. In Canvas, just now I was seeing something weird with some weird black boxes, but I'm glad to see that they seem to be coming out okay on your end. So this is a typical cartoon of a gel where you will see these empty boxes on top. Those represent the wells where the samples originated. And then you will see these bands that correspond to groups of molecules of DNA that are all the same size and that run all of them about the same distance. They run more or less at the same speed, some of them slightly faster, some of them slightly more slowly, but they traveled about the same distance, and then they remain together as a pack that we call a DNA band or a gel band. Okay? Often in these depictions of agarose gels, you will see that on either side or both, there's going to be these, we call these ladders, and these are special samples that we load on purpose. These are mixtures of DNA of known molecular sizes, of known of mixtures of fragments of known size that we can use as a reference. So if we know that that band there corresponds to a 500 base pair piece, that band to a 600, and that band to a 700 base pair piece, we now know that the fragments that we're seeing from our samples are roughly between 600 and 700, probably a 650 or so. Yeah? So once again, and based on what we were discussing before, generally when you're looking at the gel from the point of view of the wells, the smaller a DNA fragment, the faster it will migrate through the pores, and the further it will go in during that electrophoretic run than bigger fragments that are going to be dragging behind. So back to our original question, how can we use PCR to determine if a given sequence of interest is present in a sample or not? We subject that sample to a PCR reaction with primers that should detect that sequence of interest that we're chasing, in a way. And then we let the PCR reaction proceed, and we load those PCR reactions in these wells. We let the samples run for a little while, and if the PCR reaction works, there should be millions and millions and millions of copies of the same fragment that are going to migrate together, and that we should be able to see easily when we stain these gels with these fluorescent chemicals, because there is, once again, billions, if not trillions of copies of these fragments. They will all pick up this fluorescent molecule, and when we hit the gel with UV, they will emit all of that light that we can easily see. If the sequence of interest that we were after was not present in the sample, PCR reaction won't work, and we won't really see much, because all of that, a large amount of DNA that was present in the genomic background of our sample is distributed, has been fragmented into many different fragments of different sizes, and it's not like it's not there. It's just that we don't see it because it doesn't concentrate in one place. Okay? So it's much more distributed, and we simply can't see much of it. All right? So, once again, suppose that you'd like to find out if this particular given sequence is present in your sample. You would subject it to a PCR with primers A and B, and then you run your samples. And let's imagine for a second that this is what we see. We had a negative control, a sample that we know for a fact doesn't have this sequence of interest. We need to run that control to make sure that if the sequence of interest is not there, there is no PCR amplification. We have our positive control, which is the opposite, a sample that we know contains that sequence of interest, and we use it to make sure that our PCR works if the sequence of interest is there. And then we have our question mark, our incognito samples, 1 through 4, that we'd like to know if they contain the sequence of interest or not. So based on these results, which samples would you say contain the sequence of interest? 2 and 4 show bands that are of the same size as our positive control. Question number 1 for you, and it's not on this slide, but I just thought of it. Can we be sure that these samples 2 and 4 contain our sample of or sequence of interest? The answer is that we can be reasonably confident, and what we would do in real life with a result like this is to say, well, I might want further confirmation by ways of perhaps sequencing these samples to make sure that the fragments that I'm picking up by PCR are the real thing, and it's not just coincidence, because it could be coincidence that your samples 2 and 4 did not have your sequence of interest, but the primers that you use just happen to pick up an amplicon of about the same size. Somewhat unlikely, but not impossible. So what we would do is we would be slightly more confident to perhaps take samples 2 and 4 and subject them to the next round of verification, which might involve more sophisticated techniques, more expensive techniques, more time-consuming techniques like sequencing, okay? So the fact that a PCR amplicon is the same size as your positive control is not 100% reassurance that you're dealing with your sequence of interest, but it provides some sort of support for that conclusion. What happened in sample 3? So you're saying that one of the primers bound its target sequence, but the other one didn't? In a different location. I like that better. The original idea that maybe one of them found its target site and the other one didn't, so PCRs were starting from only one of the primers. When we think about how PCR works, the PCR would not have worked, would not have gone through that exponential accumulation, because you always need at some point each of these beyond picons to be amplified in the opposite direction to get that. One template will give you 2, 2 will give you 4, 4 will give you 8, and so forth, right? So you always need, for these degree of amplification, you always need both primers binding. But you elaborated on that idea and you said, well, maybe they're binding differently and they're just further apart from one another and they're amplifying an amplicon that is much longer, right? Now, are they still binding to your sequence of interest, or they're binding somewhere else? It might include the sequence of interest, or they might be binding somewhere else. When we say that we design primers against our sequence of interest, we try and be as specific as we can. There are programs out there that will tell you, if you're working with a given species, any species of your choice, if you tell it what primers you'd like to use, those programs will tell you what are the chances that they may bind elsewhere in the genome. And you pick the primers that are the most specific to your site of interest as possible. But that is not to say that it may not partially bind to other places. And if it were to only partially bind somewhere else in the genome, and both the forward and the reverse are facing one another, then given enough time, they may pick up those, what we would call non-specific amplicons. Amplicons that were created by the PCR reaction, but that were not specific to your intended sequence of interest. The other possibility is what you mentioned, that in fact, in sample 3, primers A and B were binding where they should. It's just that the sequence between them got a little longer, right? And that may have been something that happened specifically in sample 3. 1, 2, 3 could be any samples. It could be different individuals of a species. It could be species from different locations. It could be same species after some sort of mutagenesis. The point is that you intended primers A and B to bind at specific locations, but sometimes you can't control what's between them. Because of that, not only can we use PCR coupled with gel electrophoresis to find out if a given sequence is present in a sample, but we can take it a step further and use it to find out which DNA sequence is present in a sample. And to illustrate this point, I'm going to bring you back to something that we saw when we were talking about the short sequence repeats and how these things happen when there is DNA strand slippage during DNA replication, which gives rise to these very unique family of mutations. If you guys remember, we spoke of regions of DNA that have variable length. So some individuals would have 5 repeats of these short sequence repeats. Some of them would have 7, some 9, some 12. And we spent some time talking about how we can use the different combinations that exist in the human population of these variants, which differ in the number of repeats they have, to do things like lineages, like paternity tests, like CSI kind of stuff, to try and find out if someone may be associated with a crime scene and so forth, right? Back then, we kept saying that individuals were V1, V3, or V3, V4, or V2, V3, and we made a big deal out of all of that. But we never went into the detail of how we determine when an individual is V1, V2, or V2, V3, and so forth. And now you guys are equipped to understand how we do it. We do it with PCR. What we do is we design primers that flank the regions that have variable length across individuals. So I design PCR primers that are outside of these variable length regions, these, let's say, microsatellite regions. And all I need to do then is do a PCR between these primers, and depending on how many repeats were between these primers, the type of PCR amplicons that I will get. So this is a real image of one such gel where someone did PCRs against these microsatellites. And now you can see how individual number 1 was V1, homozygote for the V1 version of it. There is only one fragment because that individual happened to have both copies with the same number of repeats. But then individual number 2, or lane number 2, corresponds to someone who was heterozygote for V1, V2. Third lane is V1, V3, and so forth. So this is actually how it works when we're trying to molecularly fingerprint individuals. We would take a sample of their blood, saliva, whatever sample we may have of them. We subject those samples to PCRs with these very special pairs of primers, and then we get typically 2 bands in these gels that will tell us what V numbers they are, right? And from there, we can build pedigrees, or we can do the paternity test, or whatever we need to do. So this is a real-life use of the fact that primers may attach to the same regions in DNA of different individuals, but they may have different lengths of sequence separating them, which you can use to your advantage. So not only do you determine whether or not a given sequence is present in a sample, but what type of sequence is present in those. Second use of PCR. I told you that you can use PCR in gel electrophoresis to find out if a sequence of interest is present, which version of a sequence of interest may be present in a sample. And we can also use it to find out how many copies of a given DNA sequence are present in a sample. Right? Sometimes, so there's a number of uses for these. 2 that are very common is, let's say that you're comparing 2 samples, water samples from 2 different locations, and you would like to know how much of a given microorganism there is in one versus the other, right? Or if frogs of a given species ever take a bath in one pond versus another, so you're going to be chasing sequences from those frogs that are specific to those frogs. So you can think of a series of ecological uses of this. And then another use that is very common for this type of approach is trying to determine how many copies of a given pathogen, either a bacterium or a parasite or virus, like the COVID virus, it may be present in a sample. Right? So that way, not only do you determine if someone is infected with a given pathogen or not, but what the pathogen load is in that individual. Okay? So if I showed you this, let's say that we run a 48 cycle PCR and measure amplicon fluorescence intensity at the end of that PCR reaction. And this is what we see. This is just, these are not, I know it looks like that, but these are not bands. This is just a graph. So in the y-axis, it's just the fluorescence intensity, how bright those bands were, if you will. Right? So we have that in the sample B, the intensity is up here. Sample A and E, which is underneath, are here. This is sample D, and down here, sample C. If I asked you, which of these do you think had the more amount of template DNA in them at the beginning of the PCR? B? Why? Highest fluorescence, right? And what did we say about the number of amplicons relative to the number of template DNA molecules in a PCR reaction last week? That's proportional, right? There was a simple multiplication. It was the number of template molecules times, you guys remember that formula? Yeah, so it was the number of templates times 2 to the power of n minus 2, n being the number of cycles in that PCR. Everybody agrees? So it's proportional. The more templates I have at the beginning, the more the intensity of those bands should be at the end. So it is super tempting to say, well, the number of amplicons should be proportional. That's the constant, proportional to the number of templates at the beginning. Except that we made some emphasis at the end of last Thursday's lecture that at some point the PCR reaction is not linear anymore, and that it saturates, and that a PCR reaction, depending on factors that are not usually under our control as human beings, it can saturate and no longer produce that double amount of amplicon molecules with every cycle. At some point, it just plateaus and it won't produce any more amplicons. So the problem is that when we compare what we normally call in jargon endpoint PCRs, when we wait till the very end, there's a really high chance that all of the samples had reached saturation. And you cannot, it's not fair to compare a bunch of reactions that may have reached saturation at different points, right? It's just not fair. It's almost like comparing the lengths of different things with a broken ruler. It's just not, we're not really measuring something that is linearly proportional to what we're interested in, which is the amount of original template DNA. So comparisons in this range of reaction saturation are unfair because the system maxed out. So what we need to do is kind of look a little earlier, right? Earlier, where the fluorescences across the samples have not reached saturation, where we're still in the linear range. And that's exactly right. That's what we need to do. We need to look at these reactions that are happening in parallel to one another before they've reached saturation. The problem is that before they reach saturation, it is kind of hard for the human eye to be quantitative about any of these. When we're looking at these lighter fluorescences, I mean, the human eye is not very, very quantitative to start with. It's kind of hard to distinguish different degrees of intensity with a naked eye to start with. But also, on top of that, if you're going for lighter fluorescences, and to be on the safe side, you go for really low fluorescences, such that you know for a fact you're going to be in the linear range, then your eyes can't see it. That's why we invented what is called qPCR, quantitative PCR, or other times you'll see it referred to as real-time PCR. I don't like that name, real-time PCR, because the acronym of that is RT-PCR. As we will see in a couple of slides, we use RT for something else. So qPCR, fortunately, it's a little more popular. Quantitative PCR, qPCR. So the way it works is, instead of letting the PCR reaction proceed through completion and then stain the DNA with some sort of fluorescent molecule such that you see that most likely saturated reaction with trillions of amplicons in it, what we do is we use special reagents that function like this. So the way this works is you, as usual, you have your template DNA, right? The one that you're, in a way, querying, the one that you'd like to learn something about. You have your primers, and then instead of having regular deoxyribonucleotides, the DATP, DCTP, DTTP, and DGTP that you would normally use, the nucleotides that we need for DNA amplification, we use chemically modified nucleotides that have a very unique ability or a very unique characteristic, and that is when they get incorporated into a growing DNA molecule by the Taq polymerase, they emit a pulse of light. Right? So it's a pulse of light that the human eye couldn't see, but that these special machines that not only do the cycles of temperature that any PCR machine would do, but also have a camera pointing at these wells where the reactions take place, these cameras can see how much fluorescence is being emitted with each cycle of amplification. So the way this works is, as the PCR starts taking off and gets into the linear range, the more template molecules you had at the beginning, the faster you will reach that linear range of fluorescence that the machine can detect and say, okay, now we're in the linear range. So what this machine essentially do is they look at all of the wells in your sample and they tell you when each of those wells reached that critical point in which they started to be linear, right? When each of the samples reached that linear range. They do that, and then what they give you at the end of the day is a series of values that have to do with the cycle number at which a given sample reached that linearity. And that's where the linearity of that formula that we were thinking about before comes into play. The more template molecules that were present in a sample, the faster that linearity will be reached. The faster this machine will be able to see all of these pulses of light coming out of those wells. The fewer, if any, of those template DNA molecules present in a sample, the longer it will take for these things to start fluorescing enough that the machine can detect them. Let's move along. So far, we touched only on that middle line there to find out whether or not a copy is there, how many copies there may be, and in some special cases, which version of a given sequence there may be, right? Now let's move on to this very other common use of PCR, and that is to find out if a gene is expressed or not, and using qPCR, how much it is expressed. But let me start with this simple question. What do we mean when we say that a gene is expressed? So the very first thing that happens to a gene, whether it codes for a protein or not, that is being expressed, it gets transcribed into RNA, right? If you guys remember, DNA gets transcribed to RNA, and then RNA in some cases gets translated to protein. So knowing that a gene is there or not is one thing. Knowing if it is expressed or not is different. A gene can be present in a sample and not be expressed. Or a gene can be present in a sample and be expressed more under certain circumstances than others. So how do we get to that question? Not by looking at the gene itself, that would be invariant. What changes as a gene is being expressed or not is the amount of RNA that is being produced from it. Can we use PCR to look at RNA? So what we do is what's shown here in this cartoon. We make use of the fact that RNA can be converted back to DNA by these special enzymes called reverse transcriptases. Right? So these are RNA polymerases that work backwards, if you will. They read RNA and write DNA. Right? So what we do is we have a sample with its DNA, and that DNA will be producing a given amount of RNA or not, depending on that sample. And then we take that RNA out of the system, reverse transcribe it to something we call cDNA. That stands for copy DNA. It's just DNA molecules that are a direct copy of the RNA that was present in those samples. And that's what we subject to PCR. So the whole idea of is a given DNA of interest present in my sample or not? We shift it to, is my cDNA of interest present in a sample or not? If the answer to that question is yes, it means that gene was being transcribed. If the answer to that question is no, it means that there was no cDNA, most likely because there was no RNA, most likely because that DNA was not being expressed. And if we use qPCR on these cDNAs, we can take it a step further and say not only whether or not a cDNA is present in a sample or not, but also how much there is of it. Because the amount of cDNA that is present in a sample is proportional to the amount of RNA that is present in a sample. Okay? This is what is normally known, you'll see this in books and talks and literature, as RT-qPCR. So the RT here stands for reverse transcriptase. Right? So it's indicating that there was this process of making cDNA out of RNA molecules. The Q is for quantitative, and the PCR, of course, for PCR. So anytime you hear something about experiments in which the researchers used RT-qPCR, what they're doing is they're extracting all of the RNAs that were present in a sample, reverse transcribing them all wholesale into a collection of cDNAs, and then subjecting that pool of cDNAs to a PCR. And depending on what the question was, the answer may be, yes, this gene is expressed or not, but if you use qPCR, you can take it a step further and ask the question, how much was it expressed? Another very common use of PCR, and this is a little bit counterintuitive, we can use PCR to introduce mutations of interest in a given DNA. So far, everything we've done with PCR is to ask the question, is this sequence present in my sample or not? How many copies? Which version? Is it expressed or not? But now we're using PCR to actually make a change in our DNA of interest. And the way this works is, let's say that we have this region of the genome that we're interested in for whatever reason, and we'd like to introduce a very specific mutation here in this particular codon. Let's suppose, obviously, that this is the region that corresponds to coding region, the protein coding region of a gene, such that instead of having ATA in the coding region, we make a mutation and we turn it into an AAA, effectively changing the coding of an isoleucine into a lysine. So how do we do this via PCR? Well, at first it may sound a little crazy, but it's pretty intuitive once you see it. So what we do is we design a special primer that targets the sequence where we would like to introduce our mutation, and we purposely change that middle T to an A. Okay? When we melt, during those few seconds at 95 degrees, the original template DNA of our sample, and we let the primers anneal at, say, somewhere around 60 degrees for a few seconds, the primer will anneal almost entirely except for that mismatch of A to A. Those two A's are not going to base pair with one another. A doesn't pair with A, right? But that mismatch won't prevent the binding of the rest of the primer. So that primer will still bind, right? Maybe a little, ever so slightly more weakly, but it will still bind. And because it binds, when we let Taq polymerase do its job, it will start the synthesis of a new DNA molecule, making what on Thursday we call the Beyond Pecan, right? So the first Beyond Pecan that is produced already has in it, by design, an A there. It was introduced in the primer that we used to make this first Beyond Pecan. That was cycle number 1. In cycle number 2, this Beyond Pecan will be the template for a reverse primer that will bind somewhere upstream. It's not shown here, but the primer will bind somewhere there, and synthesis of a new DNA will start and will continue all the way until the Taq polymerase goes through that A in the beyond pecan that is now working as a template. So Taq polymerase is reading the letters of this beyond pecan in the 3' to 5' direction, and it reads T, adds an A, reads A, adds a T, reads C, adds a G, reads G, adds a C, reads A, adds a T, and when it gets to the A that was forced there by us, it adds a T. It doesn't know that that A should not have been there. It doesn't know that that was introduced purposely, right? So it just adds the T. And at the end of cycle 2, Taq polymerase will continue until it falls off. So it falls off here. And it will leave that T. Cycle 3 and thereafter, each of these beyond pecans and the amplicons that are being produced with each cycle will have an A because it's there in the primer, or a T because every time that a Taq polymerase goes through it adds a T. So now these cycles of PCR from cycle 3 onwards, they don't know any better. They don't know that they used to be a different letter in that position. They keep adding the quote-unquote wrong letter forever. And essentially what we've done is create millions and millions and millions of copies of our sequence of interest with an error in it, except that it's an error that was on purpose. So we've introduced a mutation, a point mutation, into these sequences by PCR. Cloning. So cloning is the process by which we introduce, so far we've been, in jargon we would say lifting. So we lift sequence of interest by PCR from our samples of interest, right? From our biological samples. But at some point, we'd like to introduce specific DNA sequences of interest into a biological system to see what happens, to see what they do, right? Or in some cases, we'll take it a step further starting next week, to genetically modify an organism, right? For research purposes, or in some cases for, say, agricultural or biomedical purposes, right? So that concept of introducing a foreign piece of DNA into a biological system is what we call cloning. Right? We've made a copy of a DNA of interest into a host DNA. So let me present it with this scenario. Let's imagine that we identify the gene in mice that produces an important peptide hormone in mice mammals called leptin. That's a real peptide hormone. And we'd like to produce very large amounts of leptin, but in bacteria. So essentially what we want to do is put the sequence for mouse leptin into a bacterium such that the bacterium can start produce large amounts of leptin for us. How would you do that?
Transform the bacteria.
Transform the bacteria. Okay, and what do you mean by that? Yeah, exactly. Remember Griffith, right? How Griffith introduced, he didn't know that he was doing that because he didn't even know what was going on, but the whole idea behind Griffith's experiments was that the bacteria were being transformed because there was a certain piece of DNA that was being introduced into them, right? Yeah, exactly. Now, how would you make that process a little easier? You know that this is the leptin gene in a mouse, and you'd like to put it inside of this bacteria. What would you do first? Replicate it. So replicate as in make 1 or 2 copies? Way more. How do we make way more copies of a given sequence of interest? PCR, right? So intuitively, what we do is we amplify this leptin sequence by PCR, and then we introduce it into the bacteria. As usual, it's not as simple as we thought, right? Because there's a number of issues with just amplifying by PCR the leptin gene and simply introducing. It's easy said, not so easily done, to introduce a PCR amplicon into bacteria for a number of reasons. Reason number 1, what do you think the bacterium will think when a fragment of double-stranded DNA gets into it? It will reject it. Why? Exactly. It will think that it's being infected, right? So there's a certain moment in which the bacterium thinks, this does not belong here, so I should reject it, I should destroy it. Believe it or not, bacteria have their own immune systems because they've evolved to defend themselves against pathogens that infect them. So if we try and introduce amplicons that are simply fragments of double-stranded DNA, chances are the bacteria will aggressively reject them. Instead of introducing the PCR fragments, the amplicons by themselves, into bacteria, what we do is use cloning vectors. These are pieces of DNA that have a series of characteristics that solve some of the issues that we were discussing. First and foremost, they're circular pieces of DNA. Right? They may be smaller, they may be larger. All of these vectors commonly used in molecular biology listed here are all circular. Some of them are larger, some of them are smaller, but all circular. Why? Because these circular pieces of DNA, a bacterium doesn't see as a foreign invader. It sees as something of its own. It turns out that, remember how we said that bacterial chromosomes were circular? Right? When we described bacterial chromosomes. At the time we didn't talk about this, but it turns out that bacteria also have much smaller and yet circular pieces of DNA in them that carry genes that are of interest to them in the wild that we call plasmids, right? And as usual, us humans in laboratories hacked that natural system of plasmids and we started using them as cloning vectors. Since bacteria won't reject these plasmid DNA circles, what we do is we introduce our sequence of interest into them, and then the whole thing gets introduced into bacteria. And when the bacteria pick these plasmids up, they won't destroy them. All right, so problem number one. This thing is not violently rejected by the bacterium anymore. That was problem number 0. Problem number 1, selection of transformants. So when we subject these bacteria to the presence of these plasmids on the outside, not all of them are going to pick it up. Bacteria don't go around picking up stuff from the surroundings, right? So in fact, we need to do things to them such that some of them will pick up this DNA from the surroundings. And certainly not all of them will. A very small proportion of them will. So how do we know which ones? Here's where a concept that we call selection comes in. And it's pretty self-explanatory. What we do is we purposely select for the bacteria that now have the plasmid that we want in them. How does natural selection work? Survive. What happens to the others? They die off, right? So that's exactly what we do, except that we don't subject this to natural selection. We subject it to artificial selection. And the way we do it is, we know that bacteria die when you treat them with antibiotics. Is that what you want to say? Antibiotics, right? So if you treat bacteria with antibiotics, they die. But the trick we use is the following. These vectors that I mentioned we hack in labs include in them a sequence for a gene that can degrade that antibiotic. Right? So let's say that there is an antibiotic called ampicillin, amoxicillin, like any of those antibiotics, there can be there's usually an enzyme that can chew it up. So what we do is we introduce those genes into these plasmids, right, such that the bacteria that picked up the plasmid not only picked up our sequence of interest that was piggybacking, or if you will, getting a ride on those plasmids, but also the antibiotic resistance gene. So if we take these bacteria and we plate them on a culture plate without antibiotic, all of them will thrive and survive and divide, and we will get what is called a bacterial lawn, right? It's like this massive, gooey, nasty film of bacteria, similar to what you might see grow in rotten food. But if we have an antibiotic on that bacteria, on that culture plate, everything is going to die except the handful of the bacteria that picked up the plasmid. And they picked up the plasmid with an antibiotic-resistant gene and with your sequence of interest in it. So those are called selection markers. And the last problem that we needed to sort of solve with these vectors is the following. When a bacterium picks up a plasmid and it divides, only one of the daughters will inherit that plasmid. If those bacteria divide again, after 2 generations, you're going to have only 1/4 of the bacteria carrying that plasmid. That's a problem that we call plasmid dilution. Right? As the bacteria divide, the plasmids stay in only one of the daughters, and before you know it, after 5 or 6 or 7 rounds of division, only 1 out of 64 of your bacteria have the plasmid. So how can we get around this problem? More antibiotics so that you kill all of these. Excellent. So you're still killing the ones that are non-transformants, but are you getting more and more and more of your bacteria of interest? No. You keep killing the non-transformants, but you always have only one bacterium with your DNA of interest. That's not good enough. So a different solution. So what we do is we introduce in these vectors one of those ORI sequences. We touched on this super briefly when we talked about DNA replication. That DNA replication starts at these very specific sequences that we call ORI, for origin of replication. Easy, right? So all we do is we make sure that our vector of interest has an ORI site in them. That means that every time this bacterium is replicating its DNA to give its daughters its genetic information, it will also replicate the plasmid. So out of 1 plasmid come 2, shown here. So in this mother bacterium, there's going to be 2 plasmids, and each of the daughter cells will inherit one of them. So by introducing an ORI site into a vector, now we have vectors that can allow for selection and that will prevent plasmid dilution.